Bismillahirrahmanirrahim. Assalamu alaikum. My name is Dr. Shahid. I'm assistant professor in Department of Medical Lab Sciences, University of Lahore, Pakistan. So today we will talk about antigen processing and presentation. In this lecture, we will see how antigens are processed through the process of phagocytosis in cell and how they present through MHC class molecules to T lymphocytes for the development of immune response. Antigen is any kind of foreign particle or substance or molecule that can stimulate the production of antibodies is called antigen or immunogen. So immunogens or antigen are similar, but there is some difference in both. Immunogen, all the antigens are not immunogen in nature, but all the immunogens are antigen in nature. Antigen presenting cells, so there are different antigen presenting cells. So antigen presenting cells are specialized kind of cells which include macrophages, B lymphocytes and dendritic cells. So these antigen presenting cell can be distinguished by two properties. One is the MHC molecule class 1 and class 2. Basically there are two classes for MHC, major histocompatibility molecule. One is the class 1 molecule and other is the class 2 molecule. So other properties on which we can distinguish the antigen presenting cell is the co-stimulatory signals that is necessary for the cytotoxic T cells and helper T cells activation. So antigen presenting cell, the working principle of antigen presenting cell is that firstly it will take the antigen and it will internalize the antigen by the process of phagocytosis or endocytosis, and then it will degrade the antigen into its small antigenic peptide parts, and these parts or fragments will be displayed on the surface of the antigen presenting cell through MHC class molecules. So the T helper cell can recognize and interact with the antigen class 2 MHC molecule complex on the membrane of antigens presenting cell, leading to the initiation of the co-stimulatory by the antigen presenting cell, which activate the T helper cells, and activation of T helper cells can lead to the production of the cytokines, and these cytokines then further activate the B cells, and B cell activations can produce the antibodies. So next part of our presentation is the major histocompatibility. So MHC, basically there are the two classes of MHC. One is the class 1 and other is the class 2. So these are encoded on the genes present on the chromosome number 6. The major function of the MHC is to recognize antigens. So T helper cell can recognize the antigens combined with the class 2 molecules, where TC cell generally recognize the antigen which combine with the MHC class 1 molecules. So MHC molecule can bind to the spectrum of antigenic peptides, which is derived from the intracellular degradations of antigenic molecules. So these variable regions within the MHC class molecules called cleft. Within this cleft, the antigen peptide, the fragment of antigen, can easily be sit and presented to T lymphocyte for the production of immune response. So next part of the presentation is antigen processing and presentations. So in this part, we will see how the antigen are processed, how the whole part of antigen is converted into the fragments of antigenic peptides, and how they present to MHC class molecules on the APC membrane. So the foreign particles will be degraded into the small peptides, and these peptides will combine with the MHC class 1 and class 2 complex. This conversion of proteins into the MHC associated fragment is called antigen processing and presentations. So whether a particular antigen will be processed and presented together with class 1 or class 2 MHC molecules, it's determined by the route that the antigens take to enter a cells. So now we will talk about antigen processing and presentation routes. Basically, there are two routes for antigen processing and presentation. One is the exogenous pathway and other is the endogenous pathway. In exogenous pathway, the exogenous antigen is involved. The exogenous antigen is the antigen which is produced outside of the cells and enter the cells. Next, the endogenous antigen which is produced inside the cells, we can say the viral infected cells or cancer cells, they have their own proteins. These proteins can act as endogenous antigens. So exogenous antigen which is produced outside of the host cells and enter by the process of phagocytosis or by the process of endocytosis. So phagocytosis basically is the eating process of cells, and endocytosis can be defined as the moment of any food particles or any other particles inside the cell is called endocytosis. So antigen presenting cells, as we know that there are different kind of antigen presenting cells like macrophages, dendritic cells and B cells. So antigen presenting cell will degrade the ingested exogenous antigens into peptide fragment within the endocytic processing pathway. So MHC class 2 molecule are expressed within the endocytic processing pathway, and that peptide produced by the degradation of antigen in this pathway bind to the space, bind to the cleft within the MHC class molecule region. So basically there is some space inside the MHC class 1 or class 2 molecule where antigen can easily fit or where antigen can easily sit for the presentations to T cells. The MHC molecules bearing the peptide after that then will export to the cell surface for the recognition by the T helper cells. So basically T helper cells contain CD4 receptors. So CD is the abbreviation of the cluster of differentiation. So these are the receptors that can interact with the MHC class 2 molecules. The antigen present only on MHC class 2 molecule will be interact only and only with CD4 receptor present on Th4 cells. So in this diagram we can see that how the exogenous antigen processed in the cells. Firstly, we can see that antigen is ingested by the process of phagocytosis and endocytosis. So after ingestion of the antigens, here we have like endosomes. Endosome is an membrane bonded organelle inside the cells. It will then interact with the lysosome. Lysosome is the cytoplasmic organelle which contain the acid phosphatase and hydrolytic enzymes. These hydrolytic enzymes play a role in the degradations of antigens, the conversion of the whole antigens into the peptide fragments. These peptide fragments are called antigenic peptide fragments. Then these peptide fragments will be sit or will be adjust on the MHC class molecules, and these molecules or the complex of MHC class molecules and antigen will be exported to the surface of the APC. Then these APC will interact with the CD4 receptors on T helper cells that will induce or that will accelerate the immune response. So other pathway for the antigen processing is the endogenous pathway, in which the endogenous antigen is involved. The endogenous antigen is the antigen that is produced within the host cells. Like in the virus infected cells, as we know that as the virus attack on the cells, it takes the whole metabolic machinery of the host cells and make protein for own. So these proteins can act as an endogenous antigen. Apart from this, we know that cancer cells are the oncogenes, and oncogenes produce some oncoproteins, and these oncoproteins can act as an endogenous antigens. So in endogenous pathway, the endogenous antigens will be processed and will be fragmented into the small size, and these small size fragmented antigenic parts will be presented through MHC class 1 molecules then we can see that endogenous antigens will be degraded into the peptide fragments, and that peptide fragment will be bound with the MHC class 1 molecules within the endoplasmic reticulum. So these peptide class 1 MHC complex is then will be transported or will be exported to the cell membrane surface. So all the nucleated cell express class 1 MHC molecule. All cell producing endogenous antigen use this route to process the antigen. So all the cells which contain the endogenous antigen use the endogenous pathway for the antigen processing and presentation. So these cytotoxic T cells attack and kill cells displaying the antigen MHC class 1 complex for which their receptors are specific. So in this diagram, we can see that how the endogenous antigen are processed and how they present it on the cell surface. So we can see that in this diagram, virus particle is attacking on the cell. As the viral particle attack on cell, it will inject their genetic material inside the cells in the nucleus of the cells, of the host cells. So after injection of the genetic material inside the cells, so there is a process of transcription and translations. So transcription is the process in which the mRNA will be produced, and translation is the process in which protein will be produced. So both processes will be take place there, and viral protein will be formed. So these viral proteins will be adjust in the MHC class 1 molecule which is present in the RER-ref endoplasmic reticulum. So then this will be exported towards the Golgi complex, and the Golgi complex, these complex of the MHC class 1 along with the antigen will be budded from the Golgi complex in the form of Golgi buds or Golgi vesicles. So these will be presented on the cell surface. So this is the combination of peptide class 1 MHC complex. So the whole lecture can be summarized in this last diagram. Here we can see that it consists of 8 steps. In the first step, antigen will be internalized in the cell. It will move towards the cell, inside the cell. This antigen will be degraded through the enzymatic process in the cells, and the antigen parts will be presented on the cells. As in the second step, we have the alter cell present antigen. Both the antigens will be presented through class 1 or class 2 molecules. If the antigen will be presented to class 2 molecules, MHC class 2 molecules, then it will interact with the T helper cells. If the antigen will be presented to MHC class 1 molecule, it will be interact with the cytotoxic T cells. Here in the second step, we can see that the T cell receptor recognize antigen bonded to MHC class molecules. But in the fourth step, we can see that binding antigen MHC class activates T cells, T helper cells and TC cells. So binding of both T helper cell along with the complex of MHC molecule and antigen, it will activate the T helper cell. The activated T helper cell will produce the cytokines. So cytokines basically are the chemical messengers between the immune cells. So immune cells are correlated with each other, are coordinated with each other as the immune response. As we know that they will be coordinated with each other with the help of cytokines. So in the fifth step, we can see that the activated T helper cell will secrete cytokines, and these cytokines will activate the B cells. So here we can see that in seventh step, the B cells will be activated after attaching with the antigen. So the activated B cells, as we know that the activated B cells, as the B cell will be activated, it will be differentiate into plasma cells and memory cells. The plasma cells is for the secretion of antibodies, and the memory cells is just for memorization of the immune response when the antigen will be entering next time. But on the other hand, we can see that TC cells, the complex of MHC class 1 molecule along with the antigen and TC cell complex will be activated, and the activated cytotoxic T lymphocyte recognize and kill the altered cell surface cells. So these are all about this antigen processing and presentation. Thank you so much.